Hello everyone and welcome back to the third part of the video. In this video, we are going to talk about factors affecting reaction velocity and classification of enzymes. So starting with the substrate concentration, as substrate concentration is increased, the velocity also correspondingly increases in the initial phase, but the curves flatten afterwards, meaning that when enzyme and other factors like pH and temperature are kept constant and only the substrate concentration is being increased, then more and more substrate is being added into the reaction, then initially the reaction speed or the velocity of the reaction will increase. But after some time, when all the active site of all the enzymes present in the reaction are saturated with substrate and all the enzymes are busy in converting substrate into the products, after that the reaction velocity will not increase further, but it will remain same and the curve will be flattened. In this figure, there are two lines. One is hyperbolic curve, which is a blue line, and one is in green is the sigmoid curve. The hyperbolic curve that is in blue is shown by the enzymes that follows Michaelis-Menten kinetics, but the green one is a sigmoidal curve which is shown by the allosteric enzymes, as allosteric enzymes do not follow Michaelis-Menten kinetics. The next factor that affects the reaction velocity is enzyme concentration itself. When substrate is present in abundant amount, meaning there is a lot of substrate in the reaction and the enzyme concentration is kept on increasing the enzyme concentration when the substrate is present in abundant amount, meaning that all the substrate will not turn into product, but there will always be substrate present in the reaction until the whole observation is met. So in that situation the curve will be a straight line. The graph between the velocity and the enzyme concentration will be a straight line, representing that the reaction velocity will keep on increasing and it will not flatten. The third factor is the temperature. How temperature affects the reaction velocity? As we learned in the second video that enzymes are proteins and they act like proteins and they are also denatured by heat. So in this graph we can see that it is a bell-shaped curve. The velocity of enzyme reaction increases when temperature of the medium is increased, reaches a maximum and then falls, meaning that in this graph between the reaction velocity and the temperature in degrees Celsius, at 20 degrees Celsius you can see there is least or almost negligible activity of the enzyme, but as the temperature is being increased, the reaction velocity is also increased and it attains a peak, and this peak is attained on the optimum temperature, meaning that optimum temperature is the temperature at which the enzymes work most efficiently. But on further increase of the heat or the temperature, as you can see on the 60 degrees Celsius, 70 degrees Celsius and 80 degrees Celsius, you can see that there will be a decrease in the reaction velocity, and that is because the enzymes also are proteins and on further heating they will lose their activity. So enzymes work most efficiently at their optimum temperature, which is not too low and not too high. Next factor is the pH. How pH affect the enzyme activity? The reaction velocity. So the graph between the reaction velocity and pH is also a bell-shaped curve and maximum at optimum pH. This is also the case of the temperature. This is also like the case of the temperature, sorry. And as you see that there are three different enzymes shown in this graph with their optimum pH at different pHs, like the pepsin, which works almost 1.5 to 5 pH, sorry, 1.5 to 2 pH, and trypsin, which works most efficiently at about 5 pH, and alkaline phosphatase, which works most efficiently at about 8 or 8.5 pH. So here you can also observe that there is an optimum pH for each enzyme, and their reaction they catalyze depends upon the pH that is of the reaction. Now this graph shows the effect of substrate concentration of reaction velocity for two enzymes. In this, enzyme 1 with smaller Km and enzyme 2 with larger Km are shown. The enzyme with the smaller Km, that is the enzyme 1, will have a high affinity of enzyme for the substrate, and the larger Km enzyme will have a low affinity for the low affinity of enzyme for the substrate, meaning that it is learned from the Michaelis-Menten equations that Km is inversely proportional to the affinity. So when the Km is low, the affinity of the enzyme to the substrate will be greater, meaning that it will bind faster. But if the Km is larger, and we are talking about the same substrate here, but two enzymes, as in the case of glucokinase and hexokinase, for both of which the substrate is glucose, but glucokinase work at more concentration of glucose, meaning that it needs its Km is very high, so its affinity is very low. Now talking about the classification of enzymes, and it is done on the basis of reactions the enzyme are catalyzing. Let's zoom it. So first class is the oxidoreductases. Oxidoreductases catalyze oxidation reduction reactions such as the reaction catalyzed by the lactodehydrogenase as shown in the figure. In this reaction, one part is being oxidized and the other part is being reduced. As you can see in this reaction, the substrate, that is lactate, and NAD, which is a coenzyme for this reaction, are being oxidized and reduced to make pyruvate in the presence of lactate dehydrogenase. So these kind of reactions in which there is oxidation of uh, one part of the reaction and the reduction or in the other part of the reaction or the reactants, the reactions are called oxidoreduction reactions, and these are catalyzed by oxidoreductase enzymes. The second class of enzymes are the transferases, and its example is given serine hydroxymethyl transferase. Transferase, as their name suggests, catalyzes transfer of carbon, nitrogen, phosphate containing groups, as seen in this example, that CH2 of serine is being transferred to THF and glycine is formed. The third class of the enzymes are the hydrolases. Hydrolases catalyze cleavage of bonds by adding of water, such as shown in this figure, is given the example of urease catalyzing reaction the urease enzyme catalyzed reaction, in which urea on the action of water is converted into carbon dioxide and ammonia. But this reaction takes place in the presence of urease enzyme, which is a hydrolase. The fourth classification is the lyases. Lyases catalyze cleavage of carbon-carbon bonds, carbon-sulfur bonds, या carbon-nitrogen bonds, such as shown in this example. The pyruvate is converted into acetaldehyde in the presence of pyruvate decarboxylase, in which there is breaking of carbon-carbon bonds, and the pyruvate is 3 carbon atom, 3 carbon molecule, and the acetaldehyde is only 2 carbon molecule, and 1 carbon which was broken in the presence of pyruvate dehydrogenase is converted into CO2. The fifth classification is the isomerases. Isomerases catalyze racemization of optical or geometrical isomers, such as shown in the example. Methyl malonyl coenzyme A is converted into succinyl coenzyme A. And the sixth and the last classification is the ligases. Ligases catalyze formation of bonds between carbon and oxygen, sulfur, nitrogen, coupled to hydrolysis of high energy phosphatases, such as shown in the example. The pyruvate is converted into oxaloacetate in the presence of pyruvate carboxylase enzyme, and there is conversion of ATP into ADP. So ligases require ATP for their action, and what they do, they ligate, means they add. So this 3 carbon pyruvate is converted into 4 carbon oxaloacetate by the action of the ligase enzyme. This was the classification summary of enzymes. This is it for this video, and in the next video we will talk about the coenzymes. And if you liked the video, please do like and subscribe the channel. Thank you.